A Carnegie Institution for Science audio press release for November 24, 2010. How pathogens hijack host plants. Infestation by bacteria and other pathogens results in global crop losses of over $500 billion annually. Using fluorescence resonance energy transfer, or FRET, a sensor-based technology to identify certain proteins, a research team led by the Carnegie Institution's Department of Plant Biology has discovered a novel family of pores that transport sugar out of the plant. Because the first goal of any pathogen is to access the host's food supply to allow them to reproduce, bacteria and fungi hijack the pores to access the plant sugar for food. Without food, bacteria can't divide and cannot infect other plants. This is the first time scientists have a direct handle on controlling the food supply to pathogens, and thus they have opened a new path to prevent a wide range of crop diseases at losses, including blight in rice. The researchers looked for protein sugar transporters by screening for genes that might create pore-like activities in the cell membrane in Arabidopsis, which is a relative to mustard and is widely used for research. Mutation of the pore protein genes prevents pathogen infection, such as blight in rice plants. In the absence of a pathogen, the pore proteins, called sweets, supply sugars to the developing pollen. The researchers showed that the rice resistance gene XA13, which had originally been identified by another group of researchers, functions as a sugar pore. When production of the pore is suppressed, the plant becomes resistant to a blight bacteria. This result suggests to the researchers that the protein supplies sugar to the bug during infection. A drug that can block the activity of all sweet cousins at the same time would turn off sugar supply to a pathogen that requires its host to live, and might be a powerful new way to reduce crop losses to a wide spectrum of pathogens. Understanding this process will advance infection prevention research against a wide variety of pathogens, bacterial and fungal. The researchers found that humans and animals make a similar pore protein, which may play a role in the release of sugars from cells of the intestine, liver, testes, and mammary cells in animals, including humans. Sugar efflux has been a mystery in human and animal systems as well. The novel pore proteins in Arabidopsis have counterparts in rice, the worm C. elegans, and humans. The fact that the researchers also found a similar sweet gene in humans that mediates sugar outflow in the liver and intestine suggests that study of this gene could open up a new avenue for diabetes research. The research is published in the November 25, 2010 issue of Nature. This is John Strom for the Carnegie Institution.